What are multispecific antibodies? Welcome to Biotechnology Reviews, where we unpack the science shaping tomorrow's medicine. I'm Luke McLaughlin, and today we're diving into one of the most exciting breakthroughs in therapeutic antibody engineering, multispecific antibodies or msAbs. In the next 20 minutes, you'll learn how msAbs work, why they're transforming immunotherapy, and what the future holds for this revolutionary platform. Multispecific antibodies represent a radical leap beyond traditional monoclonal antibodies. While monoclonals bind a single epitope on a single antigen, msAbs can engage multiple targets across different antigens or even on entirely different cells. Imagine a single antibody molecule that can redirect immune cells, block multiple disease pathways, or neutralize several viral strains at once. That's the promise of msAbs. This multi-targeting capability improves treatment specificity, reduces off-target toxicity, and enhances therapeutic efficacy across oncology, autoimmune diseases, infectious diseases, and even neurodegenerative disorders. Let's rewind a bit. The concept of bispecific antibodies emerged in the 1980s with hybridoma, quadromas, but poor chain pairing and stability limited their use. The real turning point came in 2009 when catumaxomab, targeting EpCAM and CD3, became the first FDA-approved bispecific antibody. Then in 2014, blinatumomab, a bispecific T-cell engager, revolutionized treatment for B-cell acute lymphoblastic leukemia. This approval validated msAbs as a clinical reality and kicked off an innovation wave. Since then, cutting-edge technologies like knob-into-hole, CrossMab, and DuoBody have brought new levels of manufacturability and stability to the table. So what makes msAbs so special? First, efficacy. By binding multiple antigens or receptors, msAbs can overcome resistance mechanisms like antigen escape, one of the most common ways cancer fights back. Second, precision. Many msAbs can redirect T cells or NK cells straight to the tumor, bypassing immune evasion strategies. Third, they're cost-efficient. Rather than developing multiple monoclonal drugs, a single msAb can replace combination therapies, streamlining development and reducing systemic toxicity. And let's not forget, personalization. msAbs can be tailored to an individual's specific disease profile, aligning beautifully with the vision of precision medicine. msAbs come in a wide variety of structural formats. Let's break it down. B-specific antibodies like blinatumomab are the most established. Trispecifics target 3 epitopes, great for diseases with multiple escape pathways. Tetraspecifics go even further, though they're still experimental. Then we've got engineering marvels like dual variable domain antibodies or DVD-IGS tandem, scFv fragments, as seen in BiTEs and nanobody-based platforms derived from camelids, with incredible tissue penetration and manufacturability. Each format has its own strengths. Some are better for tissue penetration, others for immune engagement or receptor clustering. So, how exactly do multispecific antibodies work? 1. Bridging cells. Some msAbs physically link immune cells to cancer cells. For example, CD3 on T cells is linked to a tumor antigen, forming a cytotoxic synapse that triggers cell death. 2. Receptor clustering. Others work by clustering receptors like HER2 and HER3, inhibiting signaling or inducing degradation. 3. Pathway inhibition. msAbs can block multiple signaling pathways simultaneously, perfect for diseases like autoimmune disorders, where cytokines like IL-17 and IL-23 play overlapping roles. 4. Tumor microenvironment modulation. Some msAbs activate immune cells only in acidic tumor environments, minimizing systemic toxicity while enhancing local immune response. Producing msAbs isn't easy. You need precise heavy light chain pairing, stable folding, and consistent glycosylation. That's why CHO cells remain the gold standard, though HEK293, yeast, insect, and even plant-based systems are gaining traction. Bacterial systems like E. coli work great for fragments like BiTEs, but lack post-translational modifications. The big challenge? Scalability. msAbs often aggregate, misfold, or pair incorrectly. That's where technologies like knob into whole, cross-mab, and gene ratio vector control come in, making large-scale production feasible. Let's talk about real-world impact. In cancer, msAbs enable T-cell redirection to tumor cells, checkpoint inhibition, like dual PD-1/CTLA-4 targeting, enhanced specificity with antibody-drug conjugates. In autoimmune diseases, they block dual cytokine pathways like IL-17 and IL-23, balance TREGs and effector T-cells for long-term remission. In infectious diseases, msAbs target multiple viral epitopes, HIV, SARS-CoV-2, even influenza. Enhance immune cell recruitment. In neurodegeneration, they bind amyloid beta and tau simultaneously. Use transferrin receptor targeting to cross the blood-brain barrier. Despite all this promise, msAbs face some real obstacles. Immunogenicity. Novel formats can trigger anti-drug antibody responses. Stability. Many constructs are prone to aggregation. Off-target effects, especially in T-cell engagers, which can cause cytokine storms. Even the question of whether to use msAbs versus combination therapies is still debated. In some cases, a simple cocktail of mAbs might be more flexible, but with smarter engineering like Fc silencing, pH-dependent binding, and conditional activation domains, these hurdles are being addressed. Now for the fun part. What's next? AI is changing the game. Tools like deep learning and AlphaFold predict epitope-paratope interactions, enabling de novo antibody design in silico. Synthetic biology allows creation of entirely new antibody scaffolds with non-natural amino acids and modular domains. CRISPR is optimizing cell lines, enhancing expression, and improving glycosylation consistency. Fc engineering extends half-life and enables tumor-localized activation, critical for reducing systemic immune activation. And nanobody platforms are enabling deep tissue penetration, BBB crossing, and lower manufacturing costs. The result? Smarter, safer, longer-lasting multispecific antibodies. To wrap up, multispecific antibodies are reshaping medicine. They offer a powerful way to combine the precision of monoclonals with the complexity of combination therapy in a single molecule. From cancer to chronic inflammatory diseases, to hard-to-treat infections and neurodegeneration, msAbs are paving the way for next-generation precision therapies. But challenges remain manufacturing, immunogenicity, regulatory complexities. That said, with AI, CRISPR, and bioengineering leading the charge, the future of multispecific antibodies is not just promising, it's inevitable. That's all for today's episode of Biotechnology Reviews. If you enjoyed this deep dive, subscribe and share it with colleagues. And if you're part of the biotech industry working on antibody platforms, we'd love to hear your story. Until next time, keep questioning, keep discovering.